that arrives today. I'm very happy to introduce our speaker today, Sophie Veigl, who comes for us from Vienna. Unfortunately, she's still in Vienna, and we are now connecting through the whatever kind of electron, whatever way the electrons or photons are connecting us today. But she will come to Tel Aviv soon, we hope. Very much, as you know, these days are not the regular ones. And for this, and Sophie she's coming to us as a postdoctoral fellow in the program for the history and philosophy of life sciences in the inter-university program, which Ehud is one of the directors, the representative of the Cohn Institute, and he will also talk today with Sophie on research. Sophie has finished her dissertation in Vienna, where she also actually, if I'm correct, studied all her studies. She studies biology or immunology and history and philosophy of science. And that was, as you said, in history and philosophy of biology, she did also a dissertation, and what we will hear today is connected to her work and the future work she'll do in Tel Aviv. And so today it is an occasion to know Sophie now virtually, with the hope to get to know her better in person and in different kind of discussions during the time she will spend here. As you know, it is possible, according to the current regulations, to meet people here. We don't have teaching online, but there are possibilities that a gathering of up to 10 people is possible for meetings. And I hope that will be possible in the next few weeks after you come and stay the quarantine time, which also changes a little bit from time with the regulations, but that's we can learn. Our talk today, there's the title you probably saw as I saw, is a small RNA inheritance and scientific pluralism on the role of theory in scientific controversy. She will discuss this the subject with Ehud, as I said, Ehud Lamm, which you should all know, but I will introduce for the benefit of people who perhaps do not know him and come here. Ehud teaches at our institute. He works on quite large areas of history and philosophy of biology, questions connected to psychology, to norms, to questions connected also to computer science and other related models in philosophy of science. And I didn't mention all the things, but you can get some kind of the picture. And so it's a recurrent figure in our seminar, and especially nowadays when I need your help as moderators and discussant. Ehud comes and help us much with that, and is of course currently, as I said, as a senior lecturer at our institute. So what we'll do now is, Sophie, we will start and discuss, and then Ehud will intervene and give his takes on the things. And of course, after that, we open the floor for everyone to have a discussion. So please.
Perfect. Yeah, thank you very much for this very nice introduction. I'll try to share my screen real quick for the presentation. Okay.
Yes, it works.
Does it work? Perfect.
Yeah, it works.
Good. So yeah, so again, thank you very much. I'm trying really hard to come, and for the last couple of weeks, it's always been when I thought I now have gathered everything and I can go to Israel. The embassy has always told me some other stuff is still missing or has changed. So yeah, I'm very excited to finally make it to here. but I'm also very happy that I can share with you some of my work from my PhD dissertation, which I defended in September. And particularly, I will discuss my case study, which is small RNA inheritance, and the philosophical concept I focused on, which is scientific pluralism. And I want to zoom into the question, what is the role of theory in scientific controversies? As for the structure of the talk, Ehud and I,
Sophie,
yes?
Just one moment. Change the show to
yes.
Yes.
Like that?
Yeah, yes.
Perfect. Okay. So as for the structure of the talk, so I discussed with Ehud which strategy might work best, and we decided that I will first provide some introduction to what I did and the motivation why I did it. Then I will quite briefly introduce my case study, small RNA inheritance. I won't go into big molecular details, but I will try to argue why this is a good case study for trying to answer the questions I wanted to answer. Then I will browse over materials and methods, so which type
Okay, there is some freezing. We don't hear you, Sophie. Okay. So we'll wait a few more minutes to see what happens. Oh, okay. Now you return.
Was I gone? Okay.
You were gone, yes, in the middle of the first slide.
Yes. Okay.
So and you should share again the screen,
yes.
because you are just disconnected.
Like that.
Yes, like that.
Okay, perfect. So first motivation introduction, then a brief discussion of my case study. Then I will discuss my methodology for addressing my questions and provide some results. Ehud and I agreed that this would then be a good time to start some discussion and raise some issues. And later I will move on to some analysis of my results, formulate some conclusions, ask also some questions, and then we can also discuss more generally. Okay, so first thing, so on the philosophical side, I try to answer questions with regard to scientific pluralism. And scientific pluralism, I think the last 10 or 20 years became one of the big household positions within philosophy of science, particularly within philosophy of biology. And I sometimes find it hard to find anyone who is like against scientific pluralism in one or the other way. A very general idea that motivates scientific pluralism is the idea that some or all natural phenomena might require more than one theory or more than one approach to be like fully understood. There are, of course, different versions of scientific pluralism. There are like ontological flavors of pluralism, which would be philosophers arguing because the world is of a certain kind. We also have to adapt our knowledge to this type of how the world is. There are also like more epistemological forms of scientific pluralism, which mainly argue no matter what the world is like, entertaining more than one approach, entertaining more than one theory is just a better way of gaining knowledge. While there are numerous studies about scientific pluralism and numerous case studies, when I started my PhD studies, I thought like one particular aspect about scientific pluralism hasn't had so much attention than have others. And this is, well, what do actually scientists think? What happens in a research field if it displays plurality? Is it more stable or is it unstable if more than one theory is around? Do scientists actually agree with philosophers on particular analysis? And if we are normative about our scientific pluralism, meaning if we say, well, science should become more pluralist, there should be more theories, methods, approaches be introduced, isn't it important to also see whether scientists agree with these types of analyses? Because if we look at certain types of case studies, I think like Helen Longino is a good example. So she argues, well, she wants to have more theories introduced, for example, for the study of behavior, but she also admits that the scientists that she worked with don't agree with this sentiment. So we should really ask each other, well, if we are normative pluralists, how much does our pluralism has to resonate with scientists' opinions? what I wanted to do in my dissertation is to put these questions to an empirical test. So I tried to employ social science methodology, primarily interviews, in order to answer this question. And of course, I couldn't answer it for all science, so I tried to take one case study and see which type of empirical material I could gather there. I decided on small RNA inheritance. And I did this because small RNAs are currently handled as an example for the inheritance of acquired adaptive information. What also happens a lot recently is that small RNA inheritance researchers in the articles often describe small RNA inheritance, or the phenomena they are reporting, as some indication towards some Lamarckian interpretation. Sorry. Yeah. So both by small RNA inheritance researchers, but also by evolutionary biologists, for example, defenders of the extended evolutionary synthesis, small RNA inheritance is used as evidence that neo-Darwinism does not account for all phenomena of inheritance evolution, so that some other theoretical framework might be needed in order to account, for example, for phenomena of small RNA inheritance. Thus, it seems at least that small RNA inheritance creates plurality because it seems to necessitate a different explanatory framework in order to account for all phenomena of inheritance. So very basically, what do small RNAs do? So small RNAs are very small molecules which bind to other bigger molecules in order to destroy a third kind of molecule. So in principle, small RNAs bind things and destroy them. They have been handled as very good candidate trajectories for the inheritance of acquired traits because they were shown to be able to transmit particular information, because they were shown to be adaptive, and because it was shown that at least in some model organisms, small RNA can be amplified in each generation. So one of the best studied scenarios about small RNA inheritance would be a case where, for example, the roundworm C. elegans, which is one of the most famous model organisms to study small RNA inheritance, is hit by some sort of exterior stimulus, for example, a virus attacks, and particular small RNAs are synthesized that recognize this virus and destroy viral RNAs. And these virus recognizing small RNAs can be inherited for several generations, providing some sort of immunity to future generations of C. elegans worms. And this is where people locate this sort of inheritance of acquired traits, because some sort of acquired immunity can be transmitted for several generations. So this was sort of like the starting point of my investigations. I knew that there is small RNA inheritance. It seemed from surveying the literature that people seem to propose something like another account is needed. Neo-Darwinism doesn't suffice in explaining all that there is to inheritance. And I tried to go into the field and really see what this is all about. So my methodology is on the one hand, of course, a literature survey. So I tried to read almost everything published on small RNA inheritance between, say, the years 2008 and 2018. So it's a relatively young field. The main part of my analysis focused on semi-structured interviews. So I interviewed 25 of the leading small RNA inheritance researchers. I think at the moment, they are pretty much like the important core group because small RNA inheritance is still a quite small field. And for analysis, I used a priori coding, which means I analyzed the interviews for some pre-selected themes, which I believed would be quite important to analyze. I tried to complement these things with ethnographic observations. So I spent 3 months in the lab of Odette Rechavi here in Tel Aviv, and then 3 other months in the lab of Ehud Mishka in Cambridge to see like how these things I try to work out on in the interviews play out in practice, and whether there are certain aspects of the whole situation that might be only detectable when studying the scientific practices. So the guiding question, which I also want to raise here, is the question, how does a field fare if it displays plurality? And specifically for this talk, I will focus on 2 questions that might help answer this question. On the one hand, well, how do small RNA inheritance researchers characterize their opponents? And which types of accounts of their own research field do small RNA inheritance researchers provide in interviews? Starting with the first question. So what was really curious when interviewing small RNA inheritance researchers is that they all provided a sense of that they are criticized from almost all sides, that they're up against powerful critics. But when I asked them like to specifically name who would be a critic, they never wanted to give a name, but they answered in way more general terms. So one way of addressing this was to say, well, there's an old guard of scientists who doesn't accept new stuff. They experience general hostility. They said, well, certain people don't even assess what they do, but just do not believe them. And they also characterized some sort of intellectual laziness. So they said, well, some people just don't want to bother about their results. What was more interesting was that they also managed to characterize their opponents in terms of the disciplinary orientation. So for example, they said our critics are evolutionary biologists or traditional epigeneticists or people who study transcription factors or neurobiologists or also people who tried small RNA inheritance experiments and didn't succeed. So while all of these groups of researchers who were identified as critics are quite important and interesting to talk about, I will focus on evolutionary biologists as a type of opponents. One thing that was also interesting is that interviewees sometimes identified themselves as opponents of small RNA inheritance research. So they said, well, while they believe in their own results, of course, they would say, well, there's so much crappy research within the small RNA inheritance field. So it's not that hard to be a critic and a small RNA inheritance researcher at the same time. So zooming in on the evolutionary biologists, so when I asked my interview why they think that evolutionary biologists are critics, they provided different answers. On the one hand, they thought, well, on the one hand, evolutionary biologists don't like the hype surrounding small RNA inheritance. On the other hand, they were also conscious about that evolutionary biologists didn't like the fact that molecular biologists, as small RNA inheritance researchers are, cross certain disciplinary boundaries and pretend to do something with evolution, to make claims about evolution, even though they are not trained as evolutionary biologists. The interviewees, however, agreed that they are not trained to do these things and often would say, well, you know, actually, I only want to study the mechanism. Only the mechanism attracts me. Like, I'm not trained in doing like theory-laden stuff. I'm not trained in paradigmatic thinking. And this was really in tension with how they publish things, because if they publish their results, they frame it a lot in terms of Lamarckian theory. They frame it a lot in terms of vindicating some sort of evolutionary theory. So there was a tension between like their accounts and interviews and how they write their papers. What they also said is, well, they criticized evolutionary biologists for lacking the expertise to appropriately criticize small RNA inheritance. So they said, well, evolutionary biologists never like look at a paper and criticize some sort of result, some sort of methodology, but rather they characterize this criticism as rather ideological, because it's about the question whether they like the sort of theoretical framing small RNA inheritance researchers propose or not. So first preliminary conclusion of the results is that small RNA inheritance researchers are conscious about criticisms, and there are attacks from different sides. At the same time, they're also conscious about problematic researchers within the field who they sometimes characterize as like too nutty, for example. Now, the second question is with regard to the dynamics of the small RNA inheritance research field. So what I tried to do with every researcher I interviewed was to ask them, who is part of your field? And everyone provided a different account and always a very restricted account. So I think every interviewee maybe defined 6 or 7 people as like legitimate parts of their field, while they excluded also other interviewees. When differentiating who is part of the field and who is not, they refer to a lot of different criteria. For example, they would argue that researchers working on certain model organisms are not part of the field. For example, researchers working on C. elegans would say, well, mouse people don't belong to the field because they don't do proper research, for example. Also, they would characterize the experimental setup. For example, there were people scolding others for example, using only artificial experimental systems, meaning, for example, GFP-induced systems, whereas others said, well, actually those people who use like natural setups, for example, like virus infection assays, they don't belong to the field. Also, they differentiated by the type of small RNA. So for example, people studying microRNAs would say people studying small interfering RNAs are not part of the field. Then they differentiated between inter or transgenerational results, meaning for how many generations people study small RNA inheritance. And of course, even if one researcher would check all these other boxes, if one researcher is too nutty, meaning that they are too outspoken, are too revolutionary, for example, then they also did not consider those people as part of the field. So this means there was a general concern with regards to the purity of the research field. So they were really conscious to only have a really limited number of good people, good researchers identified as part of the field. Moving on, they also identified as problematic within the research field that there are big reproducibility problems. However, if you look in the literature, small RNA inheritance researchers who criticize certain papers for being not reproducible still cite these papers. And they always said, well, these papers got something right. And because for the bigger picture that is more important, they will never like openly address these issues, which are, however, very known for the whole community, that certain results are just not true. On the other hand, while they didn't criticize people for not having reproducible results, because this was seen as a more general problem within the field, they argued that as the research field became like more cool, more en vogue, a lot of people came into the field. They characterized as free riders, so people they didn't think did good work, and so they thought that their own field is like contaminated with poor quality research. Also, they argued, well, that the type of research output they provide can be very problematic. On the one hand, they said, well, the interest by the public in epigenetic phenomena in principle can harm their field because they're not ready for the public, they would say. And they also were really suffering from one sort of like asymmetric alliance with creationists, because small RNA inheritance researchers are very often contacted by creationists. Small RNA research is often cited by creationists because what it shows is that maybe neo-Darwinism isn't all there is, and this is something creationists really like, and small RNA inheritance researchers argue that they would really suffer from this type of alliances, even though they do not reach out to creationists. In general, they also said, well, all the people that make certain Lamarckian allegations are problematic, but this was again strange because almost everyone I interviewed had certain Lamarckian allegations in their own papers. And of course, also more generally, they felt really sanctioned by orthodoxy. So they felt that all their work in reviews, when getting grants at conferences, that they are always sanctioned and held small. So as a preliminary conclusion number 2, small RNA researchers are quite concerned about guarding the boundaries of the field. They identify threats from the outside and from the inside. And I think both preliminary conclusions sort of point in the direction that the small RNA inheritance field might not be too stable. And yeah, so I would stop here and maybe go into a first round of questions and then move on later.
Thank you. Ehud, do you want to comment on that?
Yes. So thank you, Sophie. This is really interesting and a very rich paper. So I'll try to be very brief at this point so you can get more time to talk about your methods, which I think are really interesting. But since you said that everybody likes to say that they're a pluralist in the philosophy of science, let me try to be a little bit of a devil's advocate here. As you know, I find the idea, like everybody else, enticing, and the case studies, as many, not very convincing. And I think this is basically in agreement with your results. So it's not that I think we are in disagreement, but I want to push you to be more explicit about these conclusions regarding pluralism. So it seems that the RNA case is very different from the phlogiston case, for example, that is discussed by Ha-Seok Chang in his promotion of pluralism. And I really think that in retrospect, pluralism may perhaps not be the best lens through which to address what's going on. And I think that's not your problem. It's a problem of the notions of pluralism that have been developed and proposed in the literature. So I think at the end of the day, we're in agreement. But just because not everybody here is familiar with that literature, I'll use my time to give some of this introduction that maybe people are not familiar with. So Hasok Chang, for example, his idea of pluralism is that science is served by maintaining several incommensurable systems of practice, this is the term that he's using, in the same discipline and concerning the same region of nature. And the motivations are indeed normative. It's a sort of humility and prudence. It's sort of an appeal to ideas of liberal democracy and a sort of a conclusion or result of the failures of reductionism. Helen Longino has a pluralist view of pluralism. So there are various kinds of pluralism that she talks about. There's a methodological kind. There is an ontological version, like you said. And then most importantly for her, there is the social and policy conclusions that call for pluralism, which is something that I think you haven't mentioned regarding epigenetics, but of course is part of the overall discussion of epigenetics. So when you consider the things that you talked about, which show the sociological side of all of this, right? Boundary work and all that sort of thing. These notions of pluralism don't really seem to me to be very helpful. So I'll very briefly, we can talk about it later on some more, but first of all, it seems that genetics and evolutionary biology are in no relevant sense the same field of science. These are different practitioners, sometimes or often different departments, and so on and so forth. So when people from the outside, say philosophers of biology and others, say, well, look, there's pluralism going on here, this seems to be a bit stretching the evidence of what we see on the ground. The second reason that I think pluralism may be problematic when we consider things like epigenetics in general and small RNA inheritance in general is that it's not really clear what would it mean to be a pluralist about heredity. Because having multiple systems of inheritance, as for example, developed in the work of Eva Jablonka many years at the Cohn Institute, may be described as plurality, but not necessarily as pluralism, because these are different systems, they operate differently, we can study them, and so on. They only become sort of a bone of contention if you say that there is a single evolutionary theory for which they are relevant. But people doing the work, like I said, are not doing evolutionary theory. So if you start asking yourself what would be a true pluralism regarding heredity, it certainly cannot look like multiple systems of inheritance. It has to be something else. And I don't even think we know how, it's very difficult to understand what would be pluralism regarding heredity. I think your study exemplifies dynamics of scientific controversies, and that's more convincing in this case and maybe in other cases. So with this suggestion, I'll let you present how you did the study, because I think that a lot of interesting stuff is there that can help a lot of people doing research that are listening to us.
Yeah, I mean, I think also some things that you raised will come up in the latter part of the presentation. So I'll just move on. I'm sharing my screen again. Perfect. Okay, so moving on to the analysis. So I tried to work with 3 different theoretical approaches, which I will introduce in due time. One being grid group analysis established by Mary Douglas in the 1970s. The second one is the application of grid group analysis to periods of scientific controversy, which was done by David Bloor in 1978. And then I also drew a lot of insight from Mario Biagioli's work on the anthropology of incommensurability, which focuses on how social groups work in times where there is disagreement with regards to theory or even paradigms, one could say. So starting, oh yeah, and I should say I'm very happy to discuss why I use these old sources and not something newer, STS, so we can do this also later. So starting with grid group analysis. So grid group analysis hinges on the idea that the infinite array of social interactions can be sorted and classified into a few ground classes. And grid group analysis tries to account for the cultural controls within a particular group. Mary Douglas claims that actually grid group analysis can be applied to all sorts of social gatherings. So she established it in the context of anthropology, of ethnology, but she argues that actually all types of groups can be studied employing this methodology. And if one looks at case studies throughout the last 50 years, in fact, grid group analysis has been applied to a really wide range of settings. Grid group analysis actually hinges on only two characteristics of particular social groups. One is the group commitment, meaning how the different members of a group relate with each other. And the second one is grid control, meaning how hierarchical a particular system is. Having only two characteristics of groups also results in that there are 4 different configurations of social groups, according to Mary Douglas. One being the high grid high group constellation, where there is a lot of hierarchical structures, but the individuals are very nicely connected. Often it is said that a good example for high grid high group structures, for very hierarchical structures, would be, for example, the Labour Party in the UK, because you have a lot of hierarchies, you have a lot of different sections, a lot of different compartmentalization, but also a very, very strong structure that cannot be easily overthrown. The low grid high group quadrant is a quadrant where there is a lot of interconnection between the individual members of the group, but there is no, in principle, hierarchical structure in principle. So it is possible that there's some sort of charismatic leadership, but there are no rules how this leadership would work, and like age, gender, and these kind of things do not structure this group a priori. What's important about low grid high group constellations is that they are quite unstable, because like even though everything else is open for negotiations, the boundary between the outside and the low grid high group constellation is very important. It's very important to maintain it. The other two quadrants are individualism, which is low grid low group, meaning that there is no hierarchy, but also no interactions between individuals. So this is everyone for themselves, some sort of free market idea that everyone can just like try their best shot. And high grid low group structures are positions where a person either profits or has to bend because of certain hierarchies, but is not well connected to other people. And often this quadrant is characterized as one where people do not select to be, but where they're pushed into in certain situations. Now, I tried to sort of map this for the field I studied. And the most important decision was, of course, to ask where to place small RNA inheritance researchers. And because of the things I told you in the results section, the fact that their boundary work is so important, that they were so conscious in maintaining a clear boundary between inside and outside, I decided that they might be good candidates for a high group, low grid position. With regards to people they interact with, on the one hand, I think quite obviously I place the orthodoxy, the evolutionary biologists, all their critics in the high grid, high group quadrant. So this is the quadrant like where the establishment is, which is highly structured, where grants are given out, where the journals are, where big conferences are organized, and so on. In the low grid, low group quadrant, so the individualism, I place the lunatics. So as they are pushed out by small RNA inheritance researchers and they lose the group ties with small RNA inheritance, for them it's also something like everyone is up for themselves. All of them, which I tried to talk to, have really different types of ideas, so they are not homogeneously or agreeing on a similar notion of something, but they are really like all up for themselves. But which is important, they also try to access the same funding options as do small RNA inheritance researchers. And the last group would be the free riders. So the people who try to jump onto the small RNA inheritance bandwagon, but the small RNA inheritance researchers try not to let them into their reward system. So they are placed with the high grid low group because they don't have any group ties, but still try to profit from certain pre-existing structures. Well, I think the interaction between small RNA inheritance researchers and orthodoxy is quite interesting because it's like a double movement. On the one hand, small RNA inheritance researchers try to move up, they try to access funding. On the other hand, they feel a lot of pushback by orthodoxy. And with regards to this, I think it's helpful to raise a particular concept which was introduced by Mario Biagioli, who calls it strategic bilingualism, which means to decide if one is a newcomer to talk in the language of orthodoxy in order to access certain funding streams. So Biagioli argues, well, bilingualism offers a way around incommensurability. It cannot resolve it. Often, adopting a particular professional language means also to adopt a socio-professional identity. However, while members of established socio-professional groups have much to lose from becoming bilingual, bilingualism is strategically important to the invaders. So for people who are newcomers, who might speak a different language or formulate their results in different terms, in order to access certain funding streams, it makes a lot of sense to use the professional language they maybe were trained in order to access certain funding streams. So this is strategically bilingualism. And this is often what small RNA inheritance researchers do when they try to tone their stuff down in order to try to sit well sort of with orthodoxy. And I think this also accounts a lot for this movement between small RNA inheritance researchers and orthodoxy, this trying of moving up, accessing, and using this type of strategic bilingualism. A second aspect that Biagioli introduces is the question of the representation to the outside of a group versus the internal structure. And he tries to argue that particularly for a newcomer group, it is super important to present to the outside a picture of unity, consent, and homogeneity, even though they might be in principle really disparate within the group. So he, for example, takes the Copernican astronomers as an example. So he says, well, if you look at every single Copernican astronomer, they were really different in the type of theoretical framings they adopted. However, it was so important to present themselves as a group of astronomers that they tried to only negotiate these differences within the group, but presented themselves as one group unified by a certain theoretical agenda to the outside. So that's actually a really important cue for us, because like how a field appears to the outside and what we might gather if we, for example, only read research papers, and the actual disputes and theoretical commitments within the research field might be disparate. So this is something important to keep in mind. So the third preliminary conclusions are that small RNA inheritance researchers encounter substantial threats and criticisms. Sorry. They present the field as unified to the outside, which we see when we look at their written sources, probably to shield it from further criticism or to not look immature. And problems like replication issues and other things that shouldn't get out are handled within the field, and disagreement is masked if they speak to the outside. Now, the question is what else to do with the grid group frame, and that's where I went to David Bloor's work on the who tried to apply anomalies in science to the grid group framework, and he did some sort of cool merger between Imre Lakatosh's proofs and refutations and the different responses to anomaly presented in the proofs and refutations and grid group analysis. So Bloor says, well, grid group analysis particularly shows us how individuals respond to things which do not fit into the boxes and boundaries of accepted ways of thinking. The particular location within the grid group frameworks sort of defines the way a group can deal with a deviant, with a counterexample, with an anomaly. Something quite important, which Bloor also says, is that a counterexample is not a counterexample essentially or by nature, But being a counterexample is a role which is conferred upon something. So counterexamplehood, he uses the ontology of like knighthood, being made a knight is a social status entirely. Something which I want to add to Plur and his analysis, because he doesn't mention it, is that I think, well, if counterexamplehood is a social achievement, we should also expect that the recognition of something as a counterexample might not happen evenly across all grid group quadrants, because counterexamplehood is always situated, and counterexample or particular results or a particular mathematical proof that doesn't sit well with other types of claims is always produced in a particular local settings. So because counterexamples do not fly in from the outside, we should expect that different responses to anomaly and even recognizing something as an anomaly or not might be different in different parts of the grid group framework. So with regards to grid group and Plur's analysis of responses to anomaly, I think it's most worthwhile to look at the ascribed hierarchy and the factionalism, so our orthodoxy and small RNA inheritance researchers. And Plur argues that the way orthodoxy often responds to anomalies is either by monster adjustment or by exception barring. So monster adjustment would be something to say, well, if we twitch our equations a little bit, we can finally accommodate the anomaly in our framework, so it's not a problem. Exception barring is a practice to say, well, the anomalies are really strange and special causes, and actually we shouldn't deal too much with it. And this fits really nicely how, for example, evolutionary biologists sometimes respond to small RNA inheritance. On the one hand, it is often argued that, well, for example, C. elegans is such a weird model organism and doesn't represent all the other organisms, and that's why it's a weird special case. And as long as it's not more general, it shouldn't bother us too much. So this is like the exception barring example. And sometimes they argue, well, if you look at the effector molecules of small RNA inheritance, in the end, they are also encoded in DNA, and thus they are also subjected to Darwinian evolution. So this is the sort of monster adjustment. So if we look at the factors more clearly, we can sort of try to fit it into our framework without much bothering. Now, the problem is with regards to the small RNA inheritance research field, because Plur argues that within this sort of grid group location, monster barring is the particular response to anomalies. However, monster barring is a very strong response. It's just excluding something with no discussion, with no possibilities of fitting something into the realms of the social group. Now, this is weird because if small RNA inheritance is the anomaly, so to say, then it would be weird to expect that small RNA inheritance researchers monster bar small RNA inheritance. So small RNA inheritance researchers, as a matter of fact, do not regard their own subject of study as an anomaly or a monster or pathological or something like that. However, they also do not monster bar DNA inheritance. So they do not say that DNA inheritance is bad or is not true or anything like that. It's not even like both things like DNA inheritance and small RNA inheritance like coexist in their accounts. No. If we look at their practical settings, when they plan experiments, when they perform experiments, DNA inheritance and small RNA inheritance are both parts of how they plan their experiments. So in a sense, they're dualists with regards to both things. So if small RNA inheritance is not recognized as an anomaly, maybe monster barring might operate on a different level. And my idea with regards to that is that maybe there are certain monster barring techniques that prevent that small RNA inheritance can ever get the status of the anomaly. And I think, so from my interview data, there are two particular ways how small RNA inheritance monster bar. On the one hand, they bar theory within the research field and Lamarckism. So when they responded, well, I don't want to have anything to do with theory, I'm just interested in the mechanism. Our only job is to uncover mechanism, and theory has nothing to do with us. This is some very strong response of barring theories. Also, they bar researchers who are too nutty and are too evocative, for example, with regards to Lamarckism. So in a sense, we have like this particular constellation. So within the small RNA inheritance research field, theory is barred within the field. People try to avoid it. People try to focus on mechanism so that there is no dispute with regards to the theoretical framings. Also, they push out people who promote theory too much, and they classify them as lunatics. On the other hand, if you look at their papers, they use theory and also the Lamarckian framing to represent the field to the outside, because surely this type of Lamarckian framing also helps them access certain funds because it's a very popular topic. So my last preliminary conclusion is that with the case of small RNA inheritance and the debate surrounding it, theory has two contradicting roles. On the one hand, theory is mobilized as a representation to the outside and also unifies small RNA inheritance researchers, which are a really different group of people working on different model organisms with different types of experimental setup, but theory is the unifying momentum to the outside. On the other hand, theory is avoided in the inside and sometimes classified as a monster. So theory is sort of Janus phase, so it's double phase. It destabilizes and unifies at the same time. So trying to formulate some grander conclusions, the question is, what are the lessons maybe for the pluralism literature? And one thing which I find important to say is that plurality within a research field can be destabilizing. And that's of course important, because if we say, well, if there is a research field that seems to be a bit destabilized because of claims for different theories, for example, how should we formulate our normative pluralist positions if we want to contribute positively to a particular research field? Because often as philosophers of science, we want to tell science how it should be done and of course promote science and do not hinder it. And the other main lesson, I think, is the one about the ambiguous status of theory and also its rhetorical role. And this really plays out if you compare different sources of data and what can be obtained with only reading written sources and when also looking at other sources, for example, interview data. Also, what I think is quite important, an overall focus with theory as a representation also might mask other really interesting aspects of scientific pluralism that only come down in the scientific practices. So I'm at the end of my talk. Thank you very much for your attention. I'm looking forward to the discussion.
Thank you very much, Sophie. I'll give Ehud the first comments, and then we'll open to others. We'll open to your comments on Ehud and then to others.
Okay. So thank you, and like you said, you somewhat addressed some of the issues that I wanted you to say more about. Maybe now you'll have more chance to do that. Because again, it seems that you shy away from being very clear about what you think about pluralism at the end. So maybe that's a little mean, but I want to push you a little bit on this.
Okay.
But very quickly, a few remaining thoughts. But first, I just want to say that I entirely agree with your methodological conclusions. So I think a lot of philosophical as well as other discussions of science falls into the trap of imagining what's going on instead of asking and doing the empirical legwork. So that's very impressive, and your conclusion sit well with what I know from, let's call it, anecdotal data, right? Just talking to people. So it seems, so very quickly, my thoughts are that I'd like to hear your comments on are, first of all, it seems that there is a little bit of pluralist discourse rather than real pluralism. So people like to say that there are being pluralists or something, but essentially, if you work on small RNA inheritance, that's what you're working on. You're not pluralist in any respect, and the discipline is not pluralist in any respect. People are pursuing different lines of work, and they use the pluralism as a sort of, like you said, to get funding or attention or whatever. First, the second issue is that it seems that in general, when we have evolution as part of a story, a standard response is that something may happen, but it doesn't happen often enough or in important cases, what John Beatty called relative significance debates. So we don't debate whether something is possible, just whether it's important. So I wonder if what you said about exception barring and monster barring are basically techniques of relative significance debates. Essentially, we could go through Bloor, going to Douglas, or we can go through the route of John Beatty and his idea of relative significance debates, which would be interesting because, like I said, I think thinking about this as a controversy is perhaps more interesting. And the reason is that I think if we ask ourselves where this is headed, the idea is to find a better monism, right? To enlarge the monism, not pluralism, right? Again, this just doesn't, if we follow the results that you present or just the work in the field, it just doesn't seem that the direction this is going is towards pluralism. Finally, it seems that you sort of, between the lines or in a few sporadic cases, mentioned these cases, what might be called the political economy of all of this. Getting money, getting jobs, incentives, and so on. And I wonder if this is where, you know, God is in the details, right? This is where the things are. In some cases, you get more money by saying this is Lamarckian. In other cases, you're not going to get funding. And this is all much more cynical than any grand theory going on, which would also mean that the conclusions are much less generalizable. Right? It's not just, you know, these quadrants of Mary Douglas. It depends on the specific case. And because here, when we talk about epigenetic phenomena, it has implications, like you said, for creationists and others, right? The political economy is different than if this was some other field. And then this becomes interesting, but perhaps less satisfying if you're a philosopher, right? It's not generalizable. It's just a specific case. So these are sort of more larger points, but I also would be very happy if you could tell us a little bit more about the lunatics or nutties or cases or whatever you call them, because I think that's just interesting and intriguing.
Okay, so I will start with the first question to sort of be a bit more clear with regards maybe also to my position with regards to pluralism. So I have to say, like, when I started my dissertation, I also read the usual suspect books, and I was like all in for pluralism and thought it was really great and it's the best idea ever and stuff like that. But I agree that at least for this particular case, the type of pluralism I hinted at with this grand theoretical or at least explanatory pluralism is actually imported from a different discipline, which doesn't really play a lot of role within day-to-day practical settings. I think there might be much cool stuff to say, which wasn't the main focus of my dissertation, but I tried to do a little bit on that as well. How the different scientific practices that were mobilized to found the small RNA inheritance field, to found the small RNA field in general, how this might create more interesting sources of plurality, which more go to the scientific practices, looking at the different model organism, looking at the biochemistry versus genetics divide, which was also really important for the small RNA field. So yes, I think with regards to the inheritance versus evolution debate, these things are imported into the field, but as you said, sometimes strategically mobilized, and sometimes, which I also experienced, was that researchers feel the pressure from it. So they felt that they need to answer to these questions because they do this type of research, even though they weren't actually really happy to do so. Yeah. So in this sense, so one question that I didn't address here, so I asked every researcher I interviewed whether how they sympathize with pluralism in general. And then I also asked them, like, after our discussion, what they think about pluralism and small RNA inheritance. And what was really weird is that most didn't like the idea of scientific pluralism in general, but sometimes they would really express a lot of sympathy with regards to scientific pluralism, not in their field, but with regards to this theoretical debate. And it seemed to me a little bit that the pluralism here, that like adopting pluralism is more like alleviating a pressure than like really a commitment of them. And I think if there is something positive about pluralism in the field, and if it is just to alleviate pressure, then maybe there's a good reason for it to maintain it. But I would also agree that actually for this field, at least the theoretical pluralism is very rhetorical, and one shouldn't think that it's really the one that really influences scientific practice and the field, but still it sort of destabilizes the field. And maybe we should also think about that. Because I think particularly philosophers of biology often invoke this type of pluralism for these research fields, but this also does do something to the research fields. Okay, what were the other questions?
Relative significance debates was the second issue.
Yes, I mean, I completely agree. I think that over the summer I worked a lot on the extended evolutionary synthesis debate, and also with the material from there. And I think particularly in this case, the things really run in parallel. So the relative significance debate, which I think is situated in the evolutionary EES kind of thing, really is this type of exception barring and monster adjusting practice. So absolutely agree. I think that there are a lot of parallels. Yeah. Maybe with regards to the lunatics. So what is really exciting about them is that, like, if you look them up in the internet, if you visit the laboratories, if you talk with them, they are just as any other small RNA inheritance researcher. They don't do, it's like in their practical settings, they do exactly the same stuff. However, in terms of their relations with the group, they are different. They go to different conferences. They go sometimes to what is labeled lunatic conferences. So there are some bit of fringe meetings also with regards to small RNA inheritance. So it seems that they like really, when they lose the ties to the community, they also become like more forceful or even more outspoken with the ways they gain funding, because if they have lost the bounds already, they proclaim even more the Lamarckian thing, a challenge, even more challenging, and so on. Okay.
But do they sit, so do they look the same in the sense that they are in the same kinds of departments, or can you find them located in specific areas, say more fringe geographically, lower ranked universities, maybe they're older or younger, anything like that?
So I think actually they are older. I think they were mostly European, not the highest ranked universities, and some are also like not funded anymore. But this is a very small part of them. So they are not, as most of them are not actively pushed out of the scientific reward system, but yeah, their location speaks of something, that there's some type of exclusion happening.
Okay, so thank you. I think that will be for this round, and we have a few questions, and there are but the list is you can add questions to the list. There is still time. Kati, please.
Hi.
Hi.
First of all, thank you very much, Sophie. It was a very interesting talk. And I have some sort of a more historical question. And I'd like to get your ideas regarding the connection between pluralism and scientific revolutions.
Mm-hmm.
So I know that you're studying biology as a field, but I was wondering, what do you think, from what you have learned so far about scientific pluralism? Do fields that are more scientifically pluralized tend to exhibit more some sort of a framework transitions? Or in retrospective, when we look historically back, does the theories that eventually replaced old theories were considered monster barred or lunatics, or did they evolve from the mainstream?
So, I mean, this connection to the scientific revolutions is very explicitly made, for example, by Ha-Seok Chang, because his case study is, for example, the phlogiston to oxygen transition, which is often characterized as one of the scientific revolution. And he actively says, well, it was very bad that this type of revolution happens, because every time there is a revolution, we necessarily lose something. He calls this, I think, Kuhn loss. which are losing some aspects of the older system of practice, which cannot be included by the new one. And I mean, I think the question very interesting, which is I think also a question about the structure of the scientific field, like how well can they accommodate different lines of thought? Because if they can accommodate different lines of thought better, then you don't have like the revolutionary revolutions, but some sorts of streams of thought are maybe more prominent during certain times, and maybe others are more in the back, but they are not like excluded or barred, but just the plurality of different ways of doing things is easier in certain fields and not easier in other fields.
Menachem?
Yeah, thanks. Hi, Sophie. Thank
Hi.
you for a very interesting talk. I just want to push you a bit because you're kind of, well, I don't know if this is fair to say, but you're waffling between two very, very different questions. And one is the value, the normative question of pluralism. We've just finished an elections in the States, right? And now to claim that pluralism is bad because it can destabilize, thank God for destabilization. I mean, but so the normative question of pluralism is one question. This is something I've devoted quite a few years to, and part of the fruits of that is peeping over Ehud Lamm's right shoulder, just behind him. And the other question, of course, is the descriptive sociological question of the view from within, so to speak, of how does pluralism resonate within a community, not in the eyes of a sociologist or a philosopher looking from the outside. Now, looking from the outside, I very, very strongly urge you to adopt a different kind of philosophical framework. I mean, for God's sake, Lakatos, both in the scientific program stuff and both in proofs and refutations, has no interest at all in the way people, scientists, practitioners are committed to the norms of their procession, and what kind of process of dialogue and deliberation and rethinking can change that position. The cardboard images of his, he doesn't even give them names, alpha, beta, that, you know, his students in the classroom are cardboard images. And I mean, a colleague of ours, Mara Beller, who wrote about dialogism in quantum mechanics, I mean, her model, it's still not very well developed, but it's and David Bloor, I mean, the fact that he latches on to this doesn't add a dimension to it. The problems of normativity, of agency, of commitment, of the way in which dialogue can uproot commitment, can get you to rethink commitments, which you can't do by talking to yourself, and so on and so forth. Lakatos doesn't touch on this at all, and neither does David Bloor. And I think, you know, a better place to look at are philosophers who don't talk about science, but do talk about agency and normativity and so on and so on. So, I mean, so the question of the normativity of pluralism, and I think a case has to be made, because there's no way in the world a person can change his normative system, can change his mind without being exposed to an echo chamber of very, very different views. Now, the interesting question, and it is, have scientists endorsed and internalized pluralism as a norm of science? And the answer is, by and large, no. And it's interesting. But I think that if you're talking to them already, ask them, for example, not only how they characterize the idiots who don't agree with them, right? But do they credit their challenges for anything? you know, being attacked from the outside, you know, got me thinking, forced me to re-articulate some of my stuff and so on and so forth. So push them for crediting their opponents. And of course, there's an enormous temptation to keep on talking to yourself and not be bothered by people who, you know, don't share your commitments. But one way of getting at their norms would be to ask them about their ideas of pluralism in other fields. Okay, so the people arguing with you are idiots, but what would you say about mathematics? Is it good to have pluralism within mathematics, or and to allow them to try and step out of their own framework and then look back in again by looking at some other framework. But I think you're onto a fascinating topic, and yeah, it's really, really interesting. Thank you.
Thank you. I mean, I would just raise 3 quick points. The one thing, I'm not sure that you meant this, but with regards to the cardboard figures in Lakatos, I mean, they are 18th, 17th century mathematicians, right? These were people that made these assertions, and I think that's also the main focus of Bloor, who also looks. I mean, I only provided a very small glance as what he did there, but he also tried to look at these mathematicians with regards to grid group, but also like how German university systems throughout the 18th century changed. So I would say they're not that cardboard-ish.
Sophie, if I could yeah, butt in here for a second. I mean, if you look above the line and below the line in proofs and refutations, above the line, you've got cardboard images of students who have no history, no commitments, no outer world, and they're just being clever to each other. And then the sort of references down below are to flesh and blood live mathematicians about whom he says nothing, and doesn't regard them as human agents with commitments, because he's articulated them by means of the cardboard images. That's what I'm trying to say.
Okay, so the second point, I think, where you say whether, I think part of your question with regard to the normativity could also be asked in the sense of, do we need researchers to be pluralists if we as philosophers of science find it that beneficial? So is this some sort of rules of reason, right? So we don't need them actually to be pluralists as long as they maintain different ways of thinking and thereby create plurality. So this could be also a question asked. And the third thing with regards to which roles do interviews play on criticism? And I mean, one thing I think that many of them did say is that, of course, because of this type of criticism, they are more cautious. They think harder what type of things they publish, whether they try to replicate something 3 times or 5 times before they publish it, with whom to collaborate. So of course, they don't only connotate it negatively, because they also admit there is what they call like bullshit within their field. So I would defend them a little and would say, well, they actually do attach some positive things to the criticism, depending on the criticism, right? Some they find blatantly unfair and some they find productive.
Thank you. Tammy?
Yes. Hi. Thank you. Hi. For the talk was very, very interesting. And I thought it was very interesting, the whole question about the practice, how scientists look at pluralism or how they deal with such issues as pluralism. And what I wanted to ask you is, well, you showed there are 2 different issues, the theory. So there are 2 theories competing with each other, and there's the practice that scientists in their own practice, and sometimes they will use the theory into their own advantage, and sometimes they'll push the theory. And so that was interesting aspect. And you mentioned also the value aspect or the ideology. And Longino connects between the ideology, the theory, and the practice. And so sometimes the values or the background beliefs will shape the way the scientists will ask the questions, or the way they will look at different phenomena. So how do you see in your work, in your engaging with the scientist and asking those questions, and in the interviews, how these three things play together: the values, the theory, and the practice, particularly for the scientists? How do they think about it? The values and the theory, or the ideology and the theory in their practice? And if you could see differences in the way they ask the question, or the way they frame their hypotheses and models.
So, I mean, to maybe first say a little bit about like the theory and practice thing. And I think that's actually a really cool aspect, because as I said, like if a C. elegans researcher who works on small RNA inheritance performs an experiment, they have to cross the organisms, and crossing is a genetic DNA-based kind of thing. So they always have to think about that. Also, when they perform their small RNA inheritance experiments. On the other hand, and this is also interesting, there are some types of phenomena, for example, known in Drosophila, the fruit fly, which is known to be small RNA based. For example, if they are crossed a specific way, they will become sterile because of certain small RNA inheritance effects. And these things also people apply who do not believe, so to say, in small RNA inheritance, because it's methodologically necessary to do it. So that's actually really nice that in the setting of practice, these things go much better together than if they are like explored on a theoretical level. With regards to values, I mean, what I would say, it's not that surprising, but many interviewees try to formulate their vision, so to say, in Popperian terms, which often doesn't have much to do with what they do ever in their scientific life, but I think it's still super influential what they learn on one slide in the introductory course to any type of biology, that there is falsificationism and we must test all theories. And this was also some line of criticism that people are not liberal enough, because they are, you know, they don't accept certain findings, because they don't launch appropriate criticism, because they are just in disbelief. So often this type of this dogmatism in Popperian terms of not being prepared to sort of be guided by falsificationism was one way how they criticize their opponents a lot, which I think plays out a lot in the value area.
Okay, thank you. I will try to connect two questions, one of mine and one that Chaim Dueluski asked, but he wrote me that, so I try to connect somewhere the two, and they're also connected to things that Menachem and Ehud already raised. One, which I think it's from Chaim, is the relationship between the case study you talked about, the RNA, and the general claim. So if you think that the pluralism that you have in this case is something which is characterized as of all of science in general, or more for this particular case, and if so, why in this particular case than in the others? And this somewhat connected to my own worry, which is also raised by Ehud, I think, in his first comments. And this exactly which kind of pluralism you are talking about here, because the fact that we have few explanations for the same, for different kinds of inheritance, I'm not sure it's pluralism, because they can say, okay, we know that heat can flow through a few, there are different ways to heat metal. It's not a pluralism. And so, because I would say that pluralism, I would say, is that if you take Chang, is that it's either oxygen or non-phlogiston air, And if you think about it as the two of together, there is a problem. But if you allow one group to call it one way, the other group to call it another, you are in pluralism. I'm not sure I saw the same kind of thinking in your example.
Okay, so for the first question, how to extrapolate, so to say, from my case study to the more general. So I think I would see what I try to do as some sort of proof of principle. So it might have a bearing on the more general literature to raise for also other disciplines and other case studies to jump into the empirical, jump into the research fields, and to also say, well, maybe pluralism is destabilizing also in other research contexts, but I'm super open to other constellations where this really is not the case. So I'm not too dogmatically general, but I think it's a good starting point to ask maybe similar case studies, similar questions. Then for the question whether the inheritance system pluralism is a real pluralism, I think this is really interesting, because I think what you were referring to is that maybe there's only real pluralism if two things clash. And I think this is this differentiation between, do things have to be, so to say, like true alternatives in order to really formulate the pluralism? And I think particularly in the scientific pluralism in biology, a lot of people don't seem to agree. So I do not think that biology as a discipline is special, or that biological phenomena in themselves are special, that they invite a different form of pluralism, if we are to call it pluralism, where it's not really this clashing kind of things, but we just have different ways how inheritance works. We have different accounts that contribute to behavior and so on and so forth. But I think maybe regarding the disciplinary history, maybe biology became to be a discipline where these types of different approaches that don't clash with each other really are more common. But I think if there are interesting things about how these different ways of doing things, these different ways of this different theoretical outlook, if they interestingly influence, for example, scientific practices in different ways. And I think also something might change about biology, something sociological might change, and maybe one of the inheritance systems becomes an alternative to the others. So I think as of now, you're right, these things don't clash, but I think that these are sociological processes, and some things might start clashing at a certain point.
Okay. Thank you. I'll let Ehud add a few more comments. Or is there?
Yeah, so I want to go back to something I raised at the beginning that might be more relevant to address now. So I asked you what would be a true pluralist perspective on heredity, because just having multiple phenomena cannot be it. And having a grand theory in which you fix one parameter, you get genetic inheritance, you fix another, you get epigenetic inheritance, say using some general framework like the price equation or something like that. That can't be what we're after. It had to be something radical, more radical. And I think one of the better ideas about what radically different perspective on heredity would be appears in your interpretation of Lamarck, which is something you did not discuss now. So I want to give you a chance to describe your ideas or your interpretation based on small RNA inheritance of Lamarckian pools and fluids. And because I think that might give a better idea of what kind of pluralism we may have in mind.
I will interrupt here because also there is a hint that the different people you are talking about have different Lamarck. It was not, we didn't say it clearly, but it seems the case that what, and in different, perhaps also between the grant and the paper and the interview and the paper.
I agree, definitely. I mean, Lamarck sometimes just means any type of inheritance that seems weird, and sometimes it's very nicely defined. And also, I think maybe interviewees change in different situations what they actually mean by Lamarckian. So I absolutely agree. So what I try to do for small RNA inheritance is to look how this type of inheritance actually happens. And what we can see, at least in C. elegans, is that this Lamarckian idea of use and disuse really plays out nicely. So with this use and disuse kind of explanation, we really have a different explanatory framework of how we believe evolutionary change might be operated. Because for small RNAs, it was shown that...
One moment, Sophie.
Yes.
Could you explain in 2 sentences what you mean by use and disuse?
Yes.
Because not all of us are very well versed in Lamarck.
Yes. So the idea of Lamarck was like, if a particular organism uses a certain, he called it organ, a lot, then it would increase, for example, in strength or in length and whatnot. And if they would stop using a particular organ, it would decrease in strength, length, and maybe get lost entirely. And the type of inheritance with small RNA inheritance systems is one that is inherently quantitative. And this means a small RNA is not on or off. The question is always how many of which type of small RNA is being inherited in order to cause a certain effect. And with regards to the Lamarckian dimension of use and disuse, what can be shown is that, for example, if a certain virus attacks the organism, or if the organism receives a heat shock or a cold shock, populations of small RNAs change in response to that. And as long as they are used, so to say, so as long as they are part of these responses, they're being amplified, and their concentration increases at the expense of a lot of different small RNA populations, which decrease. So you really have this nice quantitative way of, through use, through the necessity of, for example, small RNAs operating the cold shock response, they increase and increase in numbers over generations. And this is a really different explanatory framework with regards to normal, like neo-Darwinian ways of accounting for how change in organisms is operated.
Thank you. I think there is another question, if I understand. Victor, you want me to ask? I think so. So I think one. He asked if the antagonism to the specific research that you talk about, the RNA inheritance, is the result of kind of a political nature regarding the implications of this particular research field.
I think this definitely is partly the case. I mean, I think the history of evolutionary biology throughout the last 100, if not more years, is deeply political. Plus the promise of epigenetics, these ideas how we can modify our offspring, how particular actions have particular results maybe for our grandchildren, which also has this other aspect of maybe liability questions. So can you blame your grandfather for smoking, for example? Is this a liability? So absolutely, I think these topics are highly politicized, and my interviews really shy away from these questions. So one really explicitly voiced, well, they do not feel that their results are open for these arenas, but yet they are always drawn into these arenas.
And in this arena, you mean the human also, or only, or also there are, I think, much easier implications regarding genetic engineering and kind of thing.
So I mean, I didn't talk much about the mammalian field and small RNA inheritance. So this is of course very live, and I also talked to a lot of small RNA inheritance researchers in mice, and all the other interviewees who do not work on mice are so cautious of accepting anything that comes from the mouse organism, particularly because it has such a high bearing, because like the representational scope of the mouse organism surely is the human. And these findings are so contentious and so thought about. And a lot of people like transition out of the small RNA inheritance field in mice because it's so hard, because it's so political. So this is a true and real pressure, which most interviewers also try to push out of the field and do not want to be associated with anything in mice.
Okay, thank you very much. I think if there are no important questions it's time for us to conclude this session. So I thank you, Sophie, very much for this interesting talk, and we learned much about RNA, heritage and pluralism, and we are waiting to see you here and learn more and have you in our discussions. And so thank you everyone for being here with us and listening, and we'll intervene again next week. So good evening.
Thank you very much.
Thank you.
Bye. Thank you.
Bye.
Bye.
Good evening. Bye.
Thank you.
Bye, Sophie.
Bye.
Thank you.